The national flagship of NIFS stands tall with its magnificent record of delivering high-quality multidisciplinary research designed for development and scientific advancement of this nation. As a former research assistant of this great institution, I am proud to salute leadership, academic staff, and non-academic staff on this occasion of NIFS 40th anniversary. Before I begin, I would like to thank the organizing committee for inviting me for this webinar series, especially Director of SCDU, Dr. Kumari Tilakaratne, and Dr. Deepa Subasinghe. My topic for today's webinar is cold plasma technology and its applications. First of all, we need to figure out what is plasma. Plasma is a Greek word which means a moldable substance or jelly. In science, we use the term plasma to define specific state of matter. Alright then, what is matter? Anything that has mass and take up space can be identified as matter. Matter is made up of tiny particles known as atoms. There are some substances that contain only one type of atom. We call them elements. Mixtures and compounds have more than one type of atoms. So is there anything that cannot be classified as matter, or anything that doesn't have mass or take up space? Yes. Examples are things like heat, light, emotion, ideas, etc. Matter can be identifiable based on their particle arrangement, energy of the particles, and the distance between them. Solid, liquid, and gases are the typical states of matter which can be seen in our day-to-day life. The bonds between the particles in a solid is strong enough to keep them tightly packed and restrict their movements only to thermal vibrations. Because of these restricted movements between the particles, solids have definite shape, volume, and high density. Liquids have no definite shape due to the weaker bonds which let the particles to slip on each other. Still, liquids have definite volume like solids. However, in gases, the particles have even higher energy, making them part each other by large distances. Gases have no definite shape or volume due to this fact and have very low density. This is an image of actual atoms taken by our Geol NeoArm atomic resolution microscope. This is a 2D structure of molybdenum disulfide, and each of these white dots is an atom. If you take a closer look, atoms are made of nucleus and an electron cloud around it. These electrons are negatively charged and nucleus is positively charged. Electrons are attached to the nucleus of an atom by the electromagnetic force. When we increase the temperature of a gas at one point, the thermal energy starts to rip off the electrons out of the atom. This is called the ionization. Now, instead of gas molecules or atoms, what we have is electrons and ions mixture or plasma. For biologists, plasma remains the blood plasma, which is the liquid where the blood cells are suspended. Physicist Irving Langmuir given the same name to plasma because in a similar way, this is an electrified fluid carrying electrons and ions. So what are the properties of plasma? A plasma is an ionized gas. So similar to a gas, plasmas also have no definite volume or shape. Gases are good insulators, but plasmas contain free electrons and charged species. So it's a good electrical conductor, similar to some liquids and solids. Also, plasmas are affected by magnetic fields. So where can we find plasma? If you look at the sky in daytime, you can see the sun. Sun is a big ball of hot plasma. If you look at night sky, you can see stars. Stars are also made of plasma like our sun. Nebulas, we call them gas clouds, but they are actually made of plasma. Some planets got rings made of plasma and we call them dusty plasma or complex plasma. Tails of comets made of plasma. Auroras are also plasma. It caused by the collision of charged particles with atoms in Earth's upper atmosphere. Auroras can be also seen on other planets which have magnetic field and an atmosphere. Examples are Jupiter and Saturn. So if I give you the number, 99.9% of the matter in the observable universe is made of plasma. Here are some examples of plasmas in our day-to-day life. Lightning. Lightning is an electrical discharge caused by imbalances between clouds and the ground or within the clouds themselves. This static discharge ionizes the air on its path and creates an arc plasma. Neon lamps and tube lights make plasma when an electrical current flows through them. Electrons of the plasma collides with atoms and transfer its kinetic energy to them and make excited atoms. These excited atoms emit photons when they reverse back to the ground state. Same principle works on plasma TVs to illuminate the display pixels. The basic plasma cutting process involves creating an electrical channel plasma. This is accomplished by a compressed gas such as oxygen, air, or inert gas, which is blown through the nozzle at high speed. An electrical arc is then formed within the gas, which ionizes some of the gas, thereby creating an electrically conductive channel of plasma. Based on the thermal equilibrium of the species, plasmas can be classified into thermal plasmas and non-thermal or cold plasmas. In thermal plasmas, ions, electrons, and other species of the plasma are in a thermodynamic equilibrium. Cold plasma or non-equilibrium plasmas is the plasma where the temperature of the individual components are different from each other. Electrons are at higher temperature and other species are at room temperature. Let me explain it by an example. Thermal plasmas are like drinking a hot tea or coffee. You can drink it from any side of the cup, or you can even use a straw and drink from the bottom. Irrespective of from where you're drinking the coffee, you feel the same temperature. Cold plasma is like a warm potato curry. The gravy could be warm enough for you to eat, but sometimes if you try to eat potato pieces, the inside of them might be more hotter and it could burn you more. So in a typical cold plasma, the gas energies are close to room temperature and electron energies are much more higher. Because of that, you can simply touch cold plasma with your bare hands. You don't even need to wear gloves. So other than ions and electrons, cold plasma also contain with reactive species and free radicals, UV and heat radiation, visible light, etc. So there's a whole range of cold plasma setups that we can use. The common thing for each of these setup is this beautiful blue-violet plasma glow. This is a schematic diagram of a dielectric barrier discharge or DBD plasma reactor. As the name suggests, DBD device consists of two metal electrodes separated by dielectric material such as quartz glass or alumina ceramic. One electrode is grounded and the other is connected to a high voltage power supply. No arcing takes place between the high voltage electrode and ground electrode due to the presence of this dielectric material between them. The system can be ignited by supplying sinusoidal or pulse power from a generator in the frequency region of 0.05 to 500 kHz. The main parameters of a DBD device are voltage, excitation frequency, and gas completion. Depending on these parameters, the output could be either glow discharge or filamentary discharge. This is a short video clip about how we operate a DBD plasma in our lab. These are two metal electrodes. Top one is connected to the power supply and the bottom one is grounded. This dielectric chamber is made of quartz. We can remove this upper lid and place our sample inside the chamber. This is the gas inlet connected to argon gas supply, and this is the gas outlet which goes into the fume hole. This is a schematic of another cold plasma device. It's called plasma jet. Unlike DBD plasma jets are capable of extending a plasma plume away from the tip of this equipment. The sample can be placed out of the plasma, and lots of biomedical applications has been developed based on plasma jets due to this advantage. Plasma jets consist of a hollow electrode and a ground electrode separated by air, which act as a dielectric material. A plasma carrier gas is continuously flows through the hollow electrode to maintain the breakdown process. Species originated with the plasma are carried away with the gas flow to the target region, which then participate to the plasma reaction processes. This is one of our plasma jet. It uses atmospheric air as the plasma carrier gas. This is an air pump. It filters the atmospheric air and feeds to the plasma nozzle, which is ignited by the current from this AC power supply. You can place your target here for the air plasma treatment, and this optical emission spectrometer connected to the computer taking live readings from the plasma source for the identification of plasma species generated. This is how the device operates. And these are thermal camera images which measure the temperature of the operating plasma. The plasma is barely visible when the thermal mode is on, which indicates the plasma temperature is very close to the room temperature. And that's my hand, and you can see my body temperature is higher than the plasma temperature. Previous slide I briefly mentioned about optical emission spectrometer. At NIFS I remember that we had an ICP-OES. ICP-OES stands for Inductive Coupled Plasma Optical Emission Spectrometer. ICP-OES comes with a built-in plasma system where we feed our sample. But in order to characterize plasma, we don't need this ICP part because we're already making the plasma. So all we need is a plain optical emission spectrometer. And the other difference is that we are using this kind of optical fiber to feed the signal directly into the spectrometer. This analyzes the light emission from the components of the plasma, such as neutral or ionized gas atoms, free radicals, and other molecules. And this is how the spectrum looks like. This one is taken during a plasma treatment into water. You can see H-alpha line indicating atomic hydrogen and hydroxyl here. So we can assume that water dissociation is taking place, and there are some nitrogen lines and oxygen lines here. This one is taken during a plasma treatment to PTSA sodium salt. So sodium gives an intense yellow signal which pretty much outshine all other peaks except these two oxygens. This is an OES spectra of argon DBD plasma. The emission spectra of the plasma is primarily dominated by these argon bands. Hydroxyl band can be also seen here, which would be originated due to the dissociation of water. The second positive system of nitrogen can also seen here in low intensity. Also oxygen lines are located here and here, blended with argon lines in 777 and 844 nanometers. Based on Scopus data, this is how the cold plasma research studies and publications increased during the last couple of decades. This is a keyword analysis of the published cold plasma related research for last 11 years. The potential applications of cold plasma can be found in various disciplines such as agriculture, medicine, material science, etc. These are some of major application areas of cold plasmas such as biomedicine, dentistry, agriculture, ozone generation, chemical synthesis, surface treatment, and coating. Wastewater from printing and dyeing is difficult to purify because of its characteristics such as large water volume, high concentration of organic pollutants, deep chromaticity, high alkalinity, changeable water quality, and complex ingredients. Cold plasma is a highly efficient and environmentally friendly method to solve this problem because it's a rich source of high energy active particles. A laboratory-built DVD reactor was used in this study to produce non-thermal plasma. Methyl violet was initially used to simulate wastewater, and on the second, wastewater was used. Both samples show potential of treating industrial water with cold plasma treatment. Cold plasmas can be used to treat plant pathogens as well. This is a green emerald leaf with fungal infection, and this is how it's been treated with a microplasma jet. After 3 weeks of plasma treatment, you can see how the spots disappear. This is the mechanism of how plasma treatment works against plant pathogens. Plasma generated species passing through the micron-stistomas in a leaf, and it weakened the function of the oil vacuoles and cell membrane of fungal cells, resulting in plasma-induced inactivation. If you treat water with plasma for a few minutes, the resulting solution is called plasma-treated water. You can't see any difference between plasma-treated water and ordinary water by its look. But my PhD supervisor used to call this water magic water or holy water. Let me explain why. In this experiment, strawberries were inoculated with Staphylococcus aureus, and controls treated with water, and 2 samples were treated with 10 minutes and 20 minutes plasma treated water. This study demonstrates plasma treated water or plasma activated water has the potential to decontaminate Staphylococcus aureus inoculated on strawberries without including significant change in color, firmness, and pH of the strawberries. This is another interesting experiment. These are 3 tomato plants and these are 3 bell pepper plants. It looks like they are in different stages of lifetime, but all these plants are 60 days old. Plants in the left are treated with tap water throughout this time. Plants in the middle and right are watered with plasma activated water for 9 days and tap water for 51 days. The other difference of the plants in the left are the seeds of them were also treated with cold plasma. When cold plasma treatment for seeds and plasma activated water treatment combined, 60% increase of the stem length is observed comparing to the control sample. This experiment shows plasma activated water has shown a significant impact on germination as well as the plant growth. This is a similar experiment to the previous one. You can freeze plasma treated water and make ice. This is an interesting comparison of tap water ice and plasma treated water ice used for seafood preservation. These are the samples treated with tap water ice and these are the samples treated with plasma activated water ice. Compared with tap water ice, plasma activated water showed significant advantage for inhibition of microbial growth and extending the storage time by 4 to 8 days. The pH of shrimps treated with plasma activated water ice remained below 7.7 during this storage. The changes in color and hardness were delayed by plasma-activated water ice treatment. In addition, plasma-activated water ice did not result in adverse changes in shrimp proteins. Therefore, this study shows that plasma-activated water ice may be used in preservation of fresh seafood. Plasma-treated water can be identified as a universal antimicrobial agent. Antimicrobial activity refers to the process of killing or inhibiting the disease-causing microbes. Various antimicrobial agents are used for this purpose. Antimicrobial may be antibacterial, antifungal, or antiviral. They all have different modes of action by which they act to suppress the infection. Nitrogen and oxygen are the main components of the atmospheric air. Atmospheric air plasma generates different types of oxygen species and nitrogen species which can proceed to several reactions. Additionally, reactive oxygen and nitrogen species forms during the plasma and liquid interactions such as superoxide, hydroxyl radical, and hydrogen peroxide are responsible for the changes of pH and antibacterial activity. Plasma-activated water kills bacteria through oxidative stress. Firstly, air plasma discharge would produce a certain amount of peroxynitrite together with other reactive oxygen and nitrogen species in the exposed water and significantly result in the acidification of the water. Peroxynitrite crossing the lipid bilayer in the cell membranes first would then initiate the lipid and protein peroxidation and nitration directly or by its decomposition to one-electron oxidants like hydroxyl ions and nitrite ions. The reaction of these particles and the cell membrane contribute to the enhancement of the permeability by the formation of transient pores in the membrane in favor of more reactive oxygen and nitrogen species penetrating into the cell. Peroxynitrite could accumulate inside cells and degrade into more highly reactive particles, resulting in an increase of intercellular reactive oxygen and nitrogen species. Both cellular apoptosis and necrosis would happen under the inducement of peroxynitrite and finally kill cells. Antibacterial mechanism of plasma jets taken place by multiple pathways. Main organic molecules found in any living organism are carbohydrates, proteins, lipids and nucleic acid. Plasma affects proteins by denaturation, amino acid oxidation and loss of enzyme activity. It also causes DNA and RNA damage and inhibits cell replication. It affects lipids and fatty acids by chemical bond breaking and radical erosion. Due to these factors, we can observe shrinkage, cytoplasm leakage, and deformation of bacterial cells. On fungi, deformation of mycelial tip due to the destruction of cellular proteins and cell apoptosis due to the DNA damage occurs, resulting loss of permeability and cell leakage. The reactive species from plasma affects the protein coat of the bacteriophage and later damage the DNA with extended time of exposure. The treatment method using cold plasma is also targeted spike proteins. The spike glycoprotein has a cysteine-rich domains and the reactive oxygen and nitrogen species present in cold plasma oxidize the cysteine. Even though the effect of reactive oxygen and nitrogen species on destructing the essential functionalities of virus are fully known, studies have shown that after a long chloroplasma exposure time, the envelope that covers the RNA genome becomes completely degraded and leads to significant modification of RNA sequence. The UV present in cold plasma also has considerable capacity in inactivating the virus by damaging their nucleic acids. Damaging of the spike proteins or the viral genomes will prevent the fusion of virus into host cells and may result in apoptosis of infected cells. Diabetes has become a worldwide pandemic and local studies shows an upward trend in Sri Lanka too. It showed a prevalence of 14.2% among males and 13.5% among females. Diabetes foot ulcers or DFU are among the most common complications of diabetes and a leading cause of hospitalization for diabetic patients. It can cause significant morbidity resulting in severe pain and poor quality of life. Approximately 15% of diabetic patients develop DFU, which precedes 84% of lower limb amputations caused by diabetes. Cold atmospheric plasma was shown to decrease bacterial load in chronic wounds. It was also presented as a novel approach to healing wounds in both in vitro and in vivo experiments. These are the results of the plasma treatment for DFU. This is the control group, a 67-year-old male patient with DFU, and this is the plasma group, a 69-year-old male patient with DFU. After 6 weeks, the wound treated with plasma is entirely healed without any signs of an infection. This is another study for cancer treatment. A plasma needle with a funnel-shaped nozzle used for this experiment. The efficiency in ablation of cultured liver cancer cells in vitro showed that oxygen addition to the argon plasma could improve the efficiency. It's found that argon, hydroxyl and oxygen radicals all formed, while the critical radicals to cell death are just oxygen and hydroxyl radicals. On a previous slide, I discussed about treating chronic ulcers by microplasma. Similarly, underwater microplasma bubbles produced by atmospheric plasma array can be used for effective biofilm dispersal. When integrated into fish tank, cold plasma generated bubbles can control bacterial biofilms on the skin of infected fish. In the picture, you can see how the symptoms disappear on biofilm infected fish skins by 15 minutes of discharge at 40 watt and prevent any new biofilm from forming. This is a video clip of the setup. You can see how these fish swim happily around this functional plasma bubble reactor. The microplasma bubble reactor is set up in a custom-made fish tank. The plasma dose is 20-second exposure twice daily. That dose is sufficient to provide reactive oxygen and nitrogen species to reduce biofilms and improve the health status of the fish. Species generated in this microplasma bubble generator include hydrogen peroxide, ozone, nitrite, nitrate, and nitric oxide. In this work, catalytically inert 2D bismuth oxide is activated by cold plasma for hydrogen evolution reaction. The tuning of the oxygen vacancy by cold plasma resulted a HCr catalyst with lower overpotential, high exchange current density, and excellent stability. This work thus paves a way to activating inherently inert metal oxide materials into high-performance electrocatalyst via cold plasma-assisted modifications. Cold plasmas can easily reduce noble metals such as silver, gold, and platinum into their nanoparticle form. Metal ion to metallic nanoparticle reduction took place due to the injected electrons from the plasma in the form of solid-lead electrons or by secondary reaction leading to the metal ion reduction. Later, diffusion and aggregation of the metal atom clusters results in nanoparticle formation. Other than aqueous solutions, ionic liquids and molten salts are also used for the plasma-assisted nanomaterial synthesis. Use of ionic liquids has advantages such as low vapor pressure and non-volatility. However, ionic liquid-based synthesis has some disadvantages too, such as the contamination of the material due to the ionic liquid decomposition. Also, solubility of the metal ions in ionic liquids is poor compared to the aqueous solutions. Transmission electron microscopy images of silver nanoparticles synthesized via DBD plasma. Resulting nanoparticles are spherical in morphology and the size of the particles are decreasing with the plasma treatment time from about 18 nanometers to 1.5 nanometers. In the TEM you can't see much about the variation of size, but DLS data you can see how the volume percentage distribution varies with the plasma treatment time. Assisted nanomaterial synthetic techniques can be also used for metal alloy nanoparticle formation. Other than the general advantages such as efficient and low temperature production, plasma-assisted bimetallic nanoparticles results in well-alloyed yield with low aggregation properties. We have conducted some preliminary experiments with silver and gold bimetallic nanoparticles and obtained some promising results. This can be continued with other metallic atoms as well, such as platinum, palladium, which can be used for many other interesting applications. Cold plasma reactions are used to synthesize non-metallic nanoparticles such as silicon nanoparticles, silicon quantum dots, germanium quantum dots, carbon-based nanoparticles such as graphene, carbon nanotubes, carbon quantum dots, carbon nanodiamonds, and fullerenes. This is a synthesis technique for switchable blue and green carbon quantum dot synthesis, which is assisted by air plasma. The starting material is an aqueous solution of pyrene tetrasulfonic acid tetrasodium salt hydrate. This technique is highly efficient, and the synthesis process can be completed within few minutes. The XPS survey spectrum shows two prominent peaks of C1s and O1s. N1s peak at 401 electron volt indicates that air plasma can dope nitrogen into the surface of carbon quantum dots in absence of any chemical nitrogen source. The atomic concentration of nitrogen is 1.55%. This is how the synthesized carbon quantum dots look under UV light. The maximum emission peak for green is peaked at 515 nanometers under the excitation of 400 nanometers. DLS data reveal the mean hydrodynamic diameter of 7.5 nanometers. According to the TEM images obtained, carbon quantum dots are spherical in shape and uniformly dispersed from each other. The average size is about 2.4 nanometers. The well-known industrial usage of cold plasma is its capacity to treat surfaces such as polymers, ceramics, composites, metal and metal alloys. Plasma treatment lacks the limitations of other functionalization techniques such as chemical and thermal treatments. These techniques are less efficient or produce byproducts which are toxic to human and the environment. Glow discharge and radiofrequency plasma treatments can activate, provide anchoring site, functionalize certain functional groups, and improve interfacial bonds. During the plasma process, chemical functional groups or free radicals on surface induce cross-linking or surface grafting. Various surface activation processes due to different plasma carrier gases are summarized in the table. This is a time of flight secondary ion mass spectrometry or TOF-SIMS comparison of tin oxide films with argon plasma and nitrogen plasma. A significant increase of nitrogen-based fragments were detected on the nitrogen plasma treated samples. Argon plasma exposed samples were also identified with some fragments at lower intensities. Spectral signatures of hydrocarbonated and oxocarbonated ions decreased with the plasma treatment. These results confirm the plasma cleaning effect on the surface. Dye-sensitized solar cells or DSSCs are a promising and increasingly cheaper alternative to the conventional silicon-based solar cells. In general, DSSCs encompass an electrode consisting of a nanocrystalline photoanode film tailored with a sensitizing dye, a platinum counterelectrode, and an electrolyte solution in between the photoanode and the counterelectrode. We use plasma-treated tin oxide photoanodes to fabricate dye-sensitized solar cells, and remarkable five-fold efficiency enhancement was achieved with the single-step surface treatment of photoanodes. The increase of the current density mainly causes the enhancement of the performance of these dye-sensitized solar cells. Therefore, it can be attributed to the increase in the number of electrons transferred from the sensitizing dye to the conduction band of the tin oxide layer. This might occur due to the increased dye absorption to the tin oxide surface as well as the elimination of trap sites in the photoanode. Tin oxide photoanodes exposed to the argon plasma show an increase of photoconversion efficiency over the controlled DSSCs. This enhancement can be attributed to the removal of the surface impurities due to argon plasma treatment. DSSCs fabricated with the photoanodes exposed to the nitrogen plasma shown the highest efficiency due to the combined effect of removal of surface impurities and the decrease of oxygen vacancies. The nitrogen plasma functionalized surface of the photoanode provide more anchorage to the sensitizing dye molecules compared to the pristine tin oxide, yielding a significant increase in the photocurrent. Additionally, plasma-treated tin oxide is capable of acting as a photocatalyst for the formation of reactive iodine species to facilitate the redox reaction of the electrolyte. A combination of all these favorable effects for the nitrogen plasma-treated photoanode significantly increase the overall photoconversion efficiencies of the fabricated DSSCs in a single-step treatment. The plasma crystal experiment conducted at the International Space Station provided new insight into an unusual type of matter known as plasma crystals. This experiment was designed to study complex or dusty plasmas which get their name from the presence of small solid particles mixed into plasma's charged gases. These particles can dramatically change the behavior of a plasma, and sometimes the particles even form crystalline structures. But they can be difficult to study on Earth because of the planet's gravity affects the way that dusty particles settle and how they crystallize. Investigations with this experiment created dusty plasmas containing argon or neon gas as well as micron-sized particles. In summary, cold plasmas can be generated under ambient conditions without the use of vacuum pumps or any other complex instruments. Energized species in the plasma can participate into various processes such as excitation, ionization, redox reactions, which can only be achieved under extreme reaction conditions if conventional methods are used. This unique property makes cold plasma an environmentally friendly, scalable and inexpensive method on broad spectrum of applications including water desalination, material synthesis, material modifications, microbial inactivation, plasma sintering, material deposition, etc. Due to these numerous advantages, cold plasma technology is a promising technology for sustainable industrial development, efficiency enhancement, and green technological processes. Before I conclude today's webinar, I would like to mention Center for Material Science at Queensland University of Technology. If you are a budding scientist, you're looking for a PhD opportunities in Australia, there are a number of openings available. Visit this URL for more details. There are few places for MPhils too. All these PhD and MPhil opportunities comes with tuition fee scholarship and living allowance. If you are lucky, you can get a relocation allowance as well, which covers your visa fee and air tickets. Also in Queensland, you won't miss Sri Lanka much because of the similar weather conditions we have here. And if you're into junk food like me, we got many delicious kottu and fried rice places here, better than Sri Lanka. So apply here if you are interested. These are my collaboration institutes: Budmore Incorporation, Australian National University, Queensland University of Technology, Condensed Matter Physics and Solid State Chemistry Laboratory of National Institute of Fundamental Studies, Sri Lanka, and James Cook University. I believe this talk will give you an idea about potential uses of cold plasma for the research areas that you are currently involved. Thank you very much for your attention. Happy 40th anniversary NIFS. Together we will move mountains tomorrow by continuing to lift stones today. Thank you once again.